So as we begin today, I want to look at this, move the microphone around where you can see me, or I can see you, and I can see the screen. And let's hope that I know how to operate this little remote. As you see here, there's a montage of Keltz's 6 species that he saw in Lake Superior. And this is actually a montage of fish that Tom Pratt and I captured up in Canadian waters in the Thunder Bay to the Nipissing Bay reach of the lake. So let's see, this will go to the next slide. And I wanted to...
I think I have the old one up.
Okay, go ahead, you can change that. While we're looking at this, these are I wanted to put the acknowledgments at the beginning because they just sort of pass by really fast. And I wanted to point out that there's a lot of people up here. And as I was looking at this, I realized I left out probably almost a half dozen of Tom Pratt's technicians that worked with us during the COVID years, where they had aged like 600 of our ciscoes, and also worked with us to do a lot of photographing of gill rakers. So we got work for everyone to do during COVID. So it takes a cast of thousands to pull something like this off. So we're ready to go again, right?
Yes.
Okay. Anyway, so that's you had a little bit to look at that list of names, and there's quite a few there. Our study begins with Kelts, and like anyone that's a student of ciscoes becomes a student of Kelts. And I remarked that Keltz's book here on the Great Lakes Corriganine fishes is really kind of like a Bible. I mean, you can talk about chapter, verse, and so on and so on. You have to go back over and over with this work. So we're guided by Kelts. And I wanted to point out that what we wanted to strive to do was to see what Kelts saw and to handle what Kelts saw and try to identify what Kelts saw in Lake Superior. And this is what he saw. So this is the 6 species that he saw in Lake Superior, and he described them. Let's see if that works. Oh, there. But we've been under the impression now for about, approaching 40 years that there really is only 4 species out there. And as Tom Pratt and I would talk, and he would say, well, that's just a Jedi mind trick. You're only supposed to see Xenithicus out there. Those other 2 do not exist. But we wanted to go back to the original. What did Kelts actually see? So our goal is to describe the morphological and genetic diversity of the ciscoes in Lake Superior broadly. And I'm not going to show you all the diversity that we know because we have a lot of data, but this is the focus on Kelts' 6. Our objective is to determine whether those 6 species are, as described by Kelts, are morphologically and genetically distinct. And our approach is to use morphology informed by genetics, and I came up with this phrase, there's a number of ways you could say this, to determine the morphological and genetic uniqueness of ciscoes. And what that means is that we begin with the genetics and let the genetics help us to understand what it is that we're seeing. It's a little different method than what we've been used to, where you typically go out and the morphologists go and measure the fish and do their analysis and generate the beautiful little graphs and then ask the geneticists to confirm my groups. We're not doing it that way. And you'll see how we've done this a little differently than has been done in the past. So we sampled broadly, and here I'm just showing you 602 fish that entered into our analysis. And these are adult fish for which we had genetic samples for. We actually sampled well over 2,000 fish over this period spread across Lake Superior, but this gets boiled down to 602 adult fish for which we were able to get useful genotypes for. So let's just step through some of the methods just so we get an idea of where we're going with all this. Again, we're considering the 6 species that Kelts had described in his monograph in 1929. The morphological characters are based on Kelts' key or diagnostic characters, and these morphological identifications are done in hand in the lab or on the ship, so that this is not something you do later. And we're using the characters of eye size, gill raker count, paired fin lengths, morphology, the jaw morphology, the body shape, and the pigmentation of the fish. Plus, we've added to this gill raker length and spacing. Morphometric measures is another way. This is after the lab work where you take the picture of the fish and you do a whole bunch of measurements from digital images. And those also include, as much as possible, Keltz's characters. As far as the post-processing goes, all the morphological characters that we use that were measured or size corrected for allometry. And that, of course, that assumes that there's going to be some changes associated with ontogeny and the size of the fish. And also, as I mentioned earlier, we restrict the analysis to adult fish, and these also are fish that have high conformation to Keltz's morphotypes for adult species. These are fish that we have a relatively high confidence in our identification. And many of you have seen this diagram of, this is Keltz's diagram here. To the left shows you the measures. I think there's 22 measures that Keltz measured on these fish. We did all of those. We have jaw morphology, sort of I call face types, and then of course a number of morphological characters we get from the gill rakers. One of the things we did with the morphological identification, again, this is the in-hand manual measurements, is we recorded our data in what we call a ciscoes scorecard, and it's basically a table with all the idealized character states for these key morphological characters across the 6 species, and these are coded as to how well they conform to those key characters, those character states. Then we can sum those and help us to identify which fish actually best matches Keltz's idealized version or morphotype for a cisco. This also gives us a permanent record where we can go back and look at what did we not get right or wrong as far as that, and so these things can be revisited and you can do some redaction. The morphometric measures, this is a common method that we've all been using for many years. This is all the measures that I mentioned that Keltz uses, plus a number of others, plus a separate analysis in which we do this. We do morphometric analysis with digital images of gill rakers. Then the genetics, and what we used was GT-sec, which is a way of generating or getting a genotype for these fish, and it is a method that's much more tolerant of degraded DNA. So that really fit well with what we had, because we didn't have the most ideal samples to work with, because this was launched during COVID and we weren't able to do any field sampling, so we had to use existing samples that we could get our hands on. So the results, we're going to start with the genetics, and we ask this question: is there a genetic basis underlying the morphological diversity of ciscoes that we sampled in Lake Superior? Put that another way, do our ciscoes cluster by groups with shared genetic identities? Can we see groups of shared genotypes? Do they exist in our data set? We're not going to ask anything about morphology yet. We're just focusing on the genetics. Do our genetic groups and clusters align with our morphological species? So we're going to go back and say, well, what did you identify those as a la Keltz, okay? So what we first get here is okay, there we go. On the left panel, we see PC, I can see it better this way, PC1 and PC2. We get segregation between 2 groups. And then we look at PC1 and PC3, we get segregation from 2 more groups, okay? In the past, we've already seen this same analysis using the same technique, and we get 3. And we know that those are associated with the most common ciscoes in Lake Superior: Ardidai, Hoyai, and Kaiai. But we came up with 4. So this is just looking at the genetics. These are the clusters, these are fish that have shared genotypes. Now, when we want to look at what those genotypes look like, they can be color-coded, and this actually we overlay onto this bar. So this has 602 genotypes arrayed from left to right. And we've used our analysis to basically group these by the clusters, numbers 1 through 4, and then they're color-coded. And you'll see over on the left, that sort of salmon-colored cluster aligns with what we've been calling Ardidai. And then hoi, what we've been calling hoi, aligns with the cluster that's green. And what we've called kai is aligning with the cluster that's blue. And then there's this pink one over there, the Rygardi, which is aligned with cluster number 4. Then you see the other 2 species, Nigrippinus and Zenithicus, which don't align with any particular cluster. They're sort of all over the place. So let's go back to this diagram again. And this time we're going to overlay this previous PCA of clusters with our species identifications. And you can see how we see clusters that are aligned with 4 different species. And I won't go into, there's some misidentifications we recognize or happen in this analysis, particularly with Zenithicus. So now we ask the question, can the genetic species be discriminated by morphology? We haven't brought the morphology into this analysis yet. So what are the morphological variables that best distinguish the 4 ciscoes that we identified and confirmed with genetics? And what is the relevance of those morphological variables? So what we did here is we have a PCA in which we use a little process called quadratic discriminant analysis. And what we ask this before we run our PCA is what are the most informative morphological variables that will maximally separate or discriminate these 4 genetic species that we've identified. And what it came up with is we have orbit size, or the eye, Gillraker length, and Gillraker spacing. And those 3 variables explain 94.2% of the variance in this data set. Of course, when I first saw this, I was taken back. I said, I've never seen a PCA explain that much variance in a data set. And each of the variants, each of the genetic species was defined by distinct clusters, as you can see. And you can see there's 95% ellipses for each of those species, and the species assignments are 97.2% correct. Now there's a couple other variables that were close runner-ups that didn't get selected, and those include Gillraker count and the pectoral fin length. And pectoral fin length in particular was basically highly correlated with eye size. You could talk to me later about what all that means. But these ecological characters, the ecological significance of these variables suggests that there's important niche segregation occurring, and this is affecting the evolution of these species. So for example, eye size is related to a spectrum from a pelagic fish with smaller eyes to a deep water fish with very large eyes that lives in a dark environment. And then you have gillrakers, which you have a combination of the length of the gillrakers and the spacing of those gillrakers, which affects the trophic specialization of the fish. So you have long, numerous gillrakers in Artidae is adapted towards a pelagic zooplankton. And then you have fish like Kai and Rygardi with fewer gillrakers, widely spaced and short, which appear to be associated with specialization on mysis and diparia. So the next question we want to ask is there concordance between morphological groups and these genetic species? Now we've asked this data set with our genetic species, tell us what those variables are. So we want to step back and ask, are there natural groupings of ciscoes based on morphology that match the genetic groups? In other words, is there any intrinsic grouping in the morphological data without imputing anything about species identification? And so we run this analysis, and it uses the same data, the same PC scores, but we're just asking it find the inherent or the clusters that occur just based on the array of these data that are in this diagram in the PC space. And what you see again, we have 95% ellipses, and the 4 clusters come out. There's the optimal number of clusters that was found was 4, and we see a very close match between these clusters and the clusters that we saw just asking the question, what are the genetic species? And we can look at this by just comparing the two presentations of these data. On the left is the genetic species, on the right is the intrinsic grouping based on just morphological similarities. And that brings us to our conclusions. And our method of morphology informed by genetics approach demonstrates that genetic differences underlie the morphological differences, and those differences, morphological differences, have implications for ecological specialization of these species. The 4 morphologically and genetically distinct ciscoes were identified, and these are Keltz's ciscoes, Cisco, bloater, Kii, and short-nosed cisco, which had not been seen before. And in a sense, what this also presents is that we have rediscovered the short-nosed cisco in Lake Superior. It was declared, essentially assumed to be distinct for nearly 40 years in the Great Lakes, so no one was expecting for us to find this, and apparently there it is. Now, Keltz's short jaw and blackfin ciscoes, from our analysis, they remain unresolved. And it's beyond the scope of this presentation to dive into what is actually going on with those two species. Our findings shed new light on the corriganine problem, and some of you may be familiar with this, and it is basically that the high level of phenotypic variation in morphology confounds our ability to delineate or identify ciscoes, and that genetics in the past has not been able to help us see differences among what we see are morphological species. This presentation shows that we've overcome this by putting these two approaches together, and with that we get some very clear delineation of these species, both genetically and morphologically. So we now look at the diversity of Lake Superior ciscoes a little bit differently than what you saw in the previous slide. We now have short-nosed cisco, and we have the blackfin and the short jaw cisco is still unresolved morphospecies. And with that, if there's any time, I can answer some questions.